we all want to be a ginkgo.
Hey everyone, this is Sophia Sanchez, a bio-developer in the making. Welcome to OBio Clips, a compilation of your favorite parts of the OBio episode. In this case, we will be hearing from Christopher Reynolds, the founder and CEO of Eden Bio, where they're using machine learning to help biotech companies have higher yields for their biotech products. So let's get started with question 1 out of 5. Tell us about Eden Bio.
We have a design, build, test loop using the machine learning, building the constructs that we predict, then testing them out. And then we have a cycle where we put the data from the tests back into the machine learning model. So we're working very hard in building a workflow where we can do high throughput, hundreds or thousands of experiments in order to generate the data we need. So the building and testing phases are key things where the latest technologies that have been developed for high throughput will allow us to do the number of builds and tests to generate the data we need.
Then I was really curious to know Chris's opinion on companies, huge companies like Ginkgo Bioworks, who are also trying to make biology easier to engineer.
Ginkgo is an inspiration to every company that comes out in space. We all want to be a ginkgo. Yay! They've obviously got areas in just research going in just about every space. So there's often talked about a rock, paper, scissors model of startups, mid-sized companies, and giant companies. And Ginkgo, in this case, is like the paper. They're like covering everything. And what a startup needs to do is be the scissors and come along and snip out a tiny bit of that paper, a niche that they can go into and excel in, and then expand outwards from that. And the rocks in these cases are the mid-sized companies which can blunt the scissors either by taking and duplicating their capabilities or just acquiring them. And then with Ginkgo, or the big giant sized companies, their papers, so they can wrap up the rocks by acquiring them. It's all an ecosystem in a way, and you need to find your place in the ecosystem, because what you don't want to be going into is just saying, okay, we want to do exactly what Ginkgo is doing and try and beat them when they have billions in resources. You need to find something that is not being looked at in much detail, and then really push forward with that.
I guess many of us have heard about the challenge in biotech and bioengineering and synthetic biology, scaling up. What does Chris think about this?
It all depends on the product. So for therapeutics, because they're such high value, you don't need much scale up, because you don't need masses of the product out in order to get a commercializable return. So something like whey, dairy protein, that's incredibly cheap. So then you need not only a big scale-up effort, because the bigger scale you have, you get economies of scale, you also need a really good scaling microorganism that's producing a lot of your product and will still be producing a lot of the product at high scale. Things that work in the lab may not work at high scale. So we're trying to minimize that by simulating high scale just right at the lab stage. So that's where we're positioning ourselves at the beginning of the value chain, where we create the strains that can then be scaled up. So we intend to leave the scale up to other companies and then take royalties from that.
Number 4, another thing we've all heard is how messy, unpredictable, and weird biology can get at times. But honestly, I'm a Gen Z, so something that I've been born with is this promise, this story of making biology easier to engineer. You know, program cells just as you program computers. That's what everyone talks about. And that's the dream, but where are we actually in terms of this?
Biology should be predictable, but it isn't, because we can't do the predictions well enough. Mm-hmm. And when AlphaFold just cracked the structure prediction problem, and when I say cracked, it still hasn't been perfected, but AlphaFold is just a vast leap over any previous predictions, and it predicts a lot of structures accurately, or even most structures. So I think there'll be similar leaps for various other parts of biology. It's just that there's more complexity in various parts of the cell. So with predicting structure, you're just predicting folds and secondary structure and tertiary structure. But with predicting how a protein is expressed in a cell, and then how it moves through the secretory pathway and then is secreted out of the cell, there's a whole conveyor belt of interactions with all different parts of the cells that all need to be considered. So I think in the future, we will have enough data to be able to predict every step of that and be able to be accurate about our predictions. It's just there's still a lot of data that needs to be collected.
Question number 5, the one that I really wanted to know the answer to. What's your advice for future biotech founders?
Is a risk, because you can form a company and it can fail, or you may not form a company, and there's so many obstacles along the way. But all of these obstacles are training. So if you stumble and fall, you will be able to try again, and this time be able to have the knowledge to avoid those or overcome those in some way. Take the leap, seize the day, and just go into business. Take a chance.
Thank you all so much for watching this video. Don't forget that you can help me by giving me a follow on Twitter at SophiaSBio, sharing this video. Listen to the episodes on the description below on Spotify, Apple Podcasts, or anywhere else you listen to your podcasts. See you in the next one.